Hi, good afternoon, everybody. This is one of these kind of architecturally determinist rooms, isn't it? You can't imagine doing many things in here, but anatomy might be one of the things that it would be possible to do. You could play Subutio or something, I guess, as well. So my name's Steve Yearley. I run the Genomics Forum. I'm director of the ESRC Genomics Policy and Research Forum. And one of the kinds of things that we do is to bring people in under our Bright Ideas fellowship scheme. So the idea is to bring people from different walks of life that are interested in genomics, whether from research or policy or legislation or from sort of pressure groups and activists and so on. And we try to organize conversations and discussions with people. But it's no exaggeration to say that this afternoon we have one of our most illustrious and brightest of our Bright Ideas fellows joining us, Bob Cook-Deegan. So I imagine the fact that many of you are here is because you know who Bob is, but just in case you've come on the basis of the advertising or whatever alone, just let me explain that Bob works really at the one of the leading genome research and policy centers, the Duke Institute for Genomic Sciences and Policy, and rather like the ambition of the Genomics Forum, what the Duke Center does is to draw together scientific research, but also with considerations about policy, legislation, justice, and some elements of social research. So Bob has been visiting here for a couple of days, did a very interesting seminar yesterday with some law students, and today he's going to be talking to us on the nicely punning, but also slightly difficult to pronounce title of ConSequences, really looking at some issues, emerging issues around genomic information. Now, Bob has trained in medicine, has an MD, but is also now very widely known for his work on policy issues around the human genome, and also for work in law, and just a fantastically broad interdisciplinary area. So I'm looking forward very much to hearing what Bob has to say. I'm concerned that there may be some people who have still got lost on the way here. There's a door downstairs that keeps being closed, and I think people keep being put off by it and going away and then coming back again. So there may be some people coming in, and there's only one way into this room. So Bob has kindly let us continue to let people in. But I think I'll hand over to Bob now, and hope you'll join with me in welcoming him in the customary way.
So this is a little scary, actually, if you think about it. I feel like, Joe, we need to come down and start our dissection, don't you think? Thank you for having me, and don't let anybody coming in. Apparently, this was something that the professors at the University of Edinburgh, when it subjected students to, if they were late to lecture for anatomy, they had to walk right in front of the professor and get fully embarrassed on the way to their chairs. I'm going to be talking about some implications of what's been happening in genomics over the last few years. And I'm going to be telling a story that weaves together two powerful technologies that have emerged in the last half century. And I'm going to be tracing some of them back to their roots, and I'm going to be making a loose analogy between what's happening in genomics now and what has happened with information technologies and telecommunication technologies on a wave that hit about 20 or 30 years ago, and looking at how those two technologies are interacting. So I'm going to talk a little bit about medical aspects of genomics, which is what most people usually think of when they're thinking about genomics, but I'm going to spend most of my time thinking about other uses of genomic information and try to foresee a little bit about how genomic information might enter your lives in a way that might seem a bit surprising. So without paying anybody, this lecture was partly set up by an advertisement that I found in the Scotsman 2 days ago. This is a picture from my iPhone. And this is a book that probably some of you, have any of you picked up this book yet? So several of you have read this book. And what it's about is using some of the technologies for identifying changes in DNA and using those to figure out lineages of descendants from human populations. And here it is directly applied to your own population. And the question here is, of course, are you a Pict or are you a Viking? Now, this is not something that I wake up in the morning thinking about most often, but some of you may, since you're swimming in the culture here in Scotland. Some of the technologies that have emerged in the last, really in the last 5 to 10 years, do seem powerful enough to be able to start mapping some aspects of human history that have been difficult to study using the other technologies that we have at hand, like archaeology, linguistics, and other ways of trying to trace human populations in their histories. So I'll come back to this in a minute, but the first thing that I need to do is salute the other folks who have been part of the team that is behind the data that I'll be showing today. The folks up in blue are the folks who had already earned their degree when they came to work at our center. The ones at the top were undergraduates at the time that they worked with us. And the ones in the yellow down here at the bottom are folks who were graduate students who have worked with us. And this is actually a very old list. It's probably, I need to do a new list, but I just haven't done that for this talk. I want to take a running start at what I'm going to be talking about. I'm not going to get to genomics immediately. We're first going to talk about general health technologies, and then we're going to talk about information technologies, and then I'll finally turn to genomics. But I want to start here. This is a slide that's got probably too much going on. It's from the graph in the upper left is a classic diagram that's from Evans and some of his collaborators, a public health diagram. And let me just walk you through it. It basically shows a curve of as you make more money moving to the right, you tend to live longer. And it shows a very strong association that's been robust over time and space that if you have more money, you live longer. And what's interesting is it's a very steep curve at the beginning. So it doesn't take a whole lot of money to increase your life expectancy. And then once you get up, it begins to get more and more expensive to earn any more to be able to pay for an increase in life expectancy. But the most remarkable thing about this graph is if you look at it, no matter how wealthy you were at the turn of the last century, you were not expected to live past your 50s. And life expectancy jumped really dramatically over the last century. So that and the graph here at the bottom that you can't see at all, but it's an absolutely beautiful graph from the World Health Organization. They don't display it this way anymore, so these data are from the year 2000. What you have is countries like Sweden and Finland and Japan on the left edge here with life expectancy over the year 80. So when you're born, you're expected to live more than 8 decades. All the way down here to sub-Saharan Africa, Botswana, Soweto, places like that, where, and this was a graph taken, these are life expectancies in the year 2000, and they're country by country. And what was happening in the year 2000 is the blue countries were getting bluer, life expectancy was increasing, and at this end, the yellow countries' life expectancy was going down, usually attributable to things like AIDS, the dissolution of political and social structure that's attributed to high levels of disease. And these are both graphs that come out of World Health Organization reports making 2 points. One is that things changed a hell of a lot in the last century. If you think about it, every week that transpired during the 20th century, there were 2 days of life expectancy added to the average human life. It's an absolutely astonishing statistic. And the other thing is that there's an incredible disparity of life expectancy across the globe, and that disparity actually was still increasing in the year 2000. Now, why is that? Well, actually, before I turn to that, let me just go through some of what this means. The main thing that it means is a human life doesn't feel anything like it did a century ago. Because these days, if you have a child, you expect that child to go to college, graduate, and have 2 or 3 careers. A century ago, you simply would not have expected that. You would have expected most of your children to die before they reached the age of 6. And they would have been dying of infectious diseases. that we would today, in most cases, be able either to prevent. Things like smallpox don't even exist anymore on the planet, or we might be able to treat them with an antibiotic or in some other way. So we don't expect our children to die. We expect to live. We expect a lot more of our bodies, and we have adjusted our expectations, and all of the economies of the developed world have changed quite dramatically over the last century in a rather remarkably short time of 3 generations. So what's that from? Well, for the first half of the last century, a lot of that change came from things that had very little to do with medical care and had very little to do with medical technology. They were things like transportation. Being able to get fresh food from one place to another is a really tremendously important technology. Sanitation, clean water is incredibly, incredibly important technology. And it's actually only at the turn of the last century that it became more and more common to have sources of clean water that were not contaminated by things like salmonella and nasty bugs that you would get when you were a young child and die. So a lot of the changes that happened at the beginning of the last century that pushed life expectancy up can be attributed to things that would have killed children that went away because of sanitation, transportation, general affluence. But interestingly, starting in the second half of the last century, technologies began to matter more and more, and all of the developed economies, what would now be the OECD countries plus some other countries, began to think systematically about applying science to the solution of the problem of disease in a really systematic way. The scale of biomedical research went up by a factor of 4 orders of magnitude in 5 decades, from World War II until the turn of the last century. Really, literally a 10,000-fold increase in the amount of money going into biomedical research. And that, of course, led to all sorts of new technologies for diagnosing disease, for treating disease, and a body of knowledge for thinking about how we do those things that changes how we think about disease and the things that we can do about it. Those technologies did turn into things like drugs for treating hypertension. Knowledge that, the idea that tobacco was bad for you to smoke because it caused lung cancer. The first papers were published here in the UK and in the US in 1950. That was new knowledge. And the intervention in that case, of course, is don't smoke. Has a huge impact on life expectancy of people who choose not to smoke. It's based on biomedical research. It was based on epidemiology, not whiz-bang technology, but it is nonetheless a result of biomedical research. But as the century progressed and as we got closer to the 80s and 90s, the scientific turning of biomedical research became more and more molecular and cellular, and more money was devoted to things that look like modern biology, where we're studying big molecules like DNA and proteins and carbohydrates, and looking at what they do within cells. And then we're looking at cells and what they do within organs, and looking at organs and how organs are organized within the body. This body of knowledge has proliferated wildly in a 5-decade period because we've gotten much more sophisticated about how we go about it. And here are the 2 revolutions that I'm going to be talking about. On the left axis, I've got a bunch of important landmarks in the development of computing and communication technologies. The first computer happened more or less at the time of the World War II. And I actually have the ENIAC computer down here listed as the first computer. We now know that that's not true. In fact, probably the first computer was on this side of the pond. But it was secret because it was being used to decode things up at Bletchley Park. So I should probably change this slide, but I am American, so I get to put ENIAC up, okay? The first commercial sale of a big computer was for doing the US Census. It was UNIVAC, was the name of the company eventually that developed that technology. And then a whole bunch of really important developments in not just the computing hardware, but in what became known as software over the years. That is, the codes that are used by computers to do useful things that we used to think about as being the sorts of things that only humans did. The first computers, in fact, were the women, mainly women, who sat around desks computing ballistic tables and decoding codes up in Bletchley Park during the Battle of Britain. These were people that were called computers. But they were doing calculations, and when it became obvious that you could start using machines to do lots and lots of calculations really quickly, and probably more accurately than the human, because they don't fall asleep, and they don't have to use pencils that break, and they don't make mistakes, because they're thinking about who they're going to date that night. Computers are better at some things than humans, and it turned out that calculations were one of those things. So it turned out that we started calling these machines that did what these women were doing. We started calling them computers. And of course, we've now got a computer that is many orders of magnitude much more powerful than the computer that I worked on when I was in high school. And I actually had the pleasure of working at a super high power, what was then called a supercomputer. It had a grand total of memory of 170K, which is, 6 orders of magnitude less than fits in this little laptop now, my MacBook Air. So a lot of things happened rather on a scale of technological advance in computing that is very hard to imagine, but we now actually take it for granted. And I'm going to be giving you a little bit of a short history of computing just because I want to lay some of the groundwork for analogies to what might be going on in genomics today. But here's on the other side of the equation here, on the genetics side, we're talking about genetics. This is a science that at the turn of the last century did not have a name. The term gene, when Mendel did all of his stuff in the 1860s, he didn't use the word gene. He talked about particles of inheritance. So what he had figured out is you could explain the inheritance of characteristics in plants by assuming that there were little things that came from mom and dad that were particles. And he knew that if you just followed the inheritance of those particles, you could explain what you observed in the progeny of the mom and the dad, and you could follow it through the generations. But he actually didn't know what the content of those particles were. And what he meant by a particle was a thing that was inherited and passed on in stable form from mom and dad to their kids. That science of studying how that works became known as genetics. Mendel's work disappeared for 35 years, was rediscovered in 3 different places at the turn of the century, exactly the turn of the last century, 1900, because suddenly it was really useful for explaining a lot of things that were hard to explain in biology. I'll fast forward through the first half of the last century of biology because I'm now going to focus on the genomic part of the story. There was a lot of debate for the first half of the last century about what are these particles of inheritance? What are they made of? If you got a particle, it's got to be made of something, right? Well, what is it? And there was a big debate, and the betting would have been, until 1944, the betting would have been that inheritance happens from these new molecules that were being studied by biochemists, the cutting-edge scientists of their day. It probably would have been proteins, right? Because they're highly variable, big molecules that contain a lot of information. Isn't that the right place that you would store genetic information? Well, that was the betting. There was this lowly molecule that's basically really, really boring. That's called the deoxyribonucleic acid. It was thought to be kind of a byproduct in cells and kind of a boring molecule. It has some sugars hanging off of it, it has some phosphates hanging off of it, and it has some base pairs hanging off of it. And lo and behold, this lowly molecule DNA turned out to be the thing that was transmitted from mom and dad to their kids and could explain what constituted these particles of inheritance. The science of studying that stuff, starting in 1953, when Watson and Crick discovered the double helical structure of DNA, became known as genetics. Progressively, starting in 1950, became more and more about studying DNA. Initially, it had been about the study of inheritance. It became about the study of the structure of DNA and how it is that the order of these base pairs that make up the DNA, the classic stair step that you can't look at a book these days. I just showed you one with a cover with the classic double helix. What's it showing? It's showing the base pairs of DNA. The code that explains the information, that stores the information for biological organisms, is stored in the order of those base pairs. And that became apparent a few years after the discovery of the double helical structure of DNA. And that led, in progression, to trying to figure out ways that you could pull the information back out of cells. And the original way of doing that was to do exactly what Mendel had done. You look at the inheritance of a characteristic in a family. So what would you look like, what would you look for in a human family? Well, the things that we care the most about are diseases. And if you wanted to use the study of DNA to explain what's going on in disease, you would actually look for a disease that's inherited in a family. So you would look for a family where there are a lot of people that get that disease. And you might be able to, if it looked like the inheritance pattern of Mendel's peas, you would know, hey, you know what? I bet if we look inside the DNA of the people that are getting the disease in this family and compare them to the people that don't get that disease in the same family, we're going to find out that they've inherited the disease from a piece of DNA that they inherited from the parent who had the disease. And it's a very, very simple-minded concept. You look for errors in spelling in the DNA that correlate with the presence or absence of disease. And we used a very biased strategy for figuring out which things to study, which is we deliberately looked for things that were inherited like the characteristics of Mendel's peas. So we looked at things like Huntington's disease that are inherited that way. And then, lo and behold, we discovered there are all sorts of other diseases, things like cancer, Alzheimer's disease, where if you look carefully, you'll actually find that there are families that have those diseases. It doesn't explain most of the cases of those diseases, but if you look for families where the inheritance pattern looks like a Mendelian inheritance pattern, you can actually study those families, and lo and behold, you can find broken genes that are associated with that disease. So then you have a really, really cool biological explanation for what's causing disease in that particular family. And that process of finding families and looking for broken genes in those families was most of medical genetics. There was a strain of population genetics and human genetics that was looking at big populations and studying other kinds of problems. But the mainstream of human genetics became medical genetics and trying to understand diseases. And that's where we were from the 1950s until the 1980s. And what began to happen in the 1980s is the second half of the equation here. Let me see if I can get my pointer over here. So we have these miraculous technologies. Are you seeing that pointer up there? Yeah. So recombinant DNA was a miraculous technology that was developed in the 1970s that basically meant you could snip a piece of DNA out of one organism and put it in another one. And it turns out DNA looks like DNA to the organism. It doesn't matter where it comes from. It's the same chemical substance. Therefore, you can take a piece of DNA from a human and grow it in a mouse or a bacterium or a yeast. It's incredibly flexible. And that allows you to move pieces of DNA around. You could take something called a gene, one of these inherited elements that's encoding something meaningful, and you move it from one organism to another, and you figure out what it does. Recombinant DNA, incredibly important technology that was developed first in the early 1970s, and then followed in close succession by a series of ways of extracting information out of DNA and actually determining the order of the base pairs that stores the biological information. So I'll be showing a little bit more of that history in a minute, so I won't go too deeply into it. But at the same time we were shifting our technological base to more and more powerful ways of studying larger and larger pieces of DNA faster, the technologies for studying DNA were getting much more powerful. And at the same time, we were developing ways of getting much more granular information at the level of DNA sequence. and beginning to use machines. So starting in the 1980s, we begin to try to determine DNA sequence not by a bunch of grad students slapping gels down and doing everything manually, but actually getting machines to read optical signals coming out of DNA molecules going through a gel, and using the mentality of engineering and instrumentation and upscale and thinking about doing things much faster, cheaper. The same mentality that was applied to silicon chips in the 1950s begins to infect the way of thinking about studying genes and DNA in human beings, and that field became known as genomics. The distinction between genetics and genomics was a distinction of three things. One is scale. You would study a lot more DNA. It was use of computing. Everything that you do in genomics requires the use of a computer because you're comparing sequences or you're comparing large molecules that you can't keep in your head. So you're using computers and you're doing things on a big scale. And the third thing is that it's high-tech whiz-bang. It takes a lot of money to be able to do this stuff. So that field became known as genomics. It really wasn't too different from genetics, except that those three things were bigger. And it's kind of a fluid term. It's almost a term like supercomputing or something like that. It moves over time. And it's not ever completely clear what it means. Where we are now is those technologies have been evolving at a remarkable pace, and I'll show you some graphs in a minute, to the point that genomics is now about, I think, to become much more important in many more people's lives than it has been. Not because it solves all the problems in the world, but because relative to other technologies, it's getting faster and cheaper, much more than the other technologies that we use. And when we have a technology that gets faster and cheaper, we start using it more and more, and we start using it for things that we never thought about using it for. And that's what I think is beginning to happen with genomics. Now, I've said I'm making an analogy between computing and genomics, but I also want to illustrate some differences. And some of these differences mean that at the end of this story, you're going to discover I don't know what's going to happen, and I suspect nobody knows exactly what's going to happen because the differences are as important as the similarities. The similarities are the speed and the power of the technology. But here are some differences. One is that in computing, while these technologies have gotten faster and cheaper, the prices have dropped rather dramatically. Now, that never happens in medicine. And it doesn't happen in medicine for a lot of reasons. But most of those reasons have to do with the way that we organize medical care. Once you get into the medical system, it's hard to get out of it. And it's sticky. And it's expensive. And the people who own and run the medical system don't want to let go of it. So medicine's got a lot more of the guild elements than most computing technologies had to contend with. I think that's going to change the way that innovation enters various kinds of markets. I'll be alluding to this a bit when I talk about direct-to-consumer genomics. The life cycles are also different. The life cycles of a technology are very different, and they are inherently different. The generation cycle for a piece of software used to be about 18 months. It's getting a little bit longer now, but it used to be about 18 months. The generation time for a new generation of computers for years was about 18 months. But very interestingly, in biomedical research, the time from discovering, making a new discovery, to making a change in the delivery of medical care has remained about 2 decades throughout all of the revolution of biomedical science over the last century. And one reason for that is you can't really short-circuit the process of studying what happens to people over time if what you want to figure out is what's going wrong in a person's body. You just can't short-circuit that. You have to follow lots of people over time and see what happens to them. It doesn't matter how much information you gather along the way, you still have to follow the time course of a human being's life. You can accelerate that in some ways that I'll allude to, but that is a fixed boundary condition in studying human biology that does not pertain to computers. I won't go over the other factors. Those are the main things that are different. Is the markets into which these technologies are introduced are medical. In the case of genomics, those are the opening markets, and that was not true for computing. Now, I'm showing this slide because I'm guessing everybody in the room knows who these two guys are. Is that true? Right? This guy is, these are two really wealthy guys. One of them's still alive. One of them isn't as of a few months ago. They are cultural icons who made a ton of money and built really big companies. And everybody knows their name. And you can't see a picture of them without seeing a picture of the companies that they created. I didn't select these photos all that carefully, right? You see that they are, they're part of our world where you have to market yourself along with your logo. My point in showing this slide is you know who these people are. They're really, really famous, and they're really, really rich. The folks who are like them, they are not the pioneers of their field. They are people who innovated in the system of delivering the applications that grew out of the computing revolution, but they are not people who created the revolution. And my point in showing this slide is I suspect that the folks who are the functional equivalent of Bill Gates and Steve Jobs are alive now, but we don't know who they are in genomics because they haven't done their work yet. Now I am going to show you some pioneers, and I'm going to do a really, really fast history of computing and telecommunications technology. These are three guys who shared the Nobel Prize for inventing the transistor. You have Bourdain, Brattain, and Shockley. Shockley became really infamous at the end of his career for his racist theories of intelligence. Bourdain is really interesting because he actually got 2 Nobel Prizes in physics. This was one for inventing the transistor. The other was for discovering superconductivity. And so all three of these guys are really famous, but I don't know very many students who I show this slide to actually know who the heck they are. And if they know anybody, they know Shockley. And if they know Shockley, it's because of what he said about IQ and race, not because of what he did early in his career. He was also kind of a jerk, so he became famous for that too. These are 2 other people that I don't know if you'll know. These are the guys who invented the chip, the integrated chip, who figured out that rather than having multiple components that you had to wire together, you could take a single semiconductor wafer and etch all of the things into it that you needed to do a lot of useful things. That was the seminal technology that gave rise to many, many things, including computers. And those are the chips that I've got in profusion in this thing and that you've got in your cell phones and everything else that does anything in our lives today in the way of communication and computing technologies. Robert Noyce was at Intel. Jack Kilby was at Texas Instruments. Kilby actually lived long enough to get the Nobel Prize and was incredibly generous in sharing credit with, these are 2 guys who actually patented their inventions independently and could have fought viciously against each other, but actually kind of liked each other and saluted each other and cross-licensed their patents so that their field could move forward, despite the pressures of having done their work in a corporate environment where the companies themselves had cross purposes. This is another person I suspect some of you in the room may know. This is Tim Berners-Lee, who, when he was working at the European Center for Nuclear Research outside of Geneva, crosses the border into France, he is the guy who figured out it would be really cool to be able to display information in a graphical way that would allow people using any sort of software to see more or less the same thing on their screen in front of them. I'm oversimplifying the technology, but he invented the hypertext markup language. And he did that so that people doing lots of expensive, high-tech experiments on really big machines that were generating lots and lots of data could share their data quickly with each other and would be able to do that despite the fact they were using different computer systems in different places. So it was the idea of interoperability and common end-user interface and coming up with a way of doing that that became known as the World Wide Web. He's also still alive and incredibly important, probably has influenced our lives as much as anybody. and yet he's not all that famous. At least most of my students don't know who he is. The final analogy I'm going to draw between genomics and computing technologies is there are some policy similarities between these two fields. I said that computers were driven initially by, I said that the first commercial computer was developed for the Census Bureau. That's a government function. The ENIAC, and of course, I alluded to Bletchley. Those are both military applications. Computers were developed for military applications. There was a very tight linkage between government uses of computational power and the development of computational technologies. In genomics, the functional equivalent is a lot of the science that has been done has been government and nonprofit funded science that has been supported by the likes of the Medical Research Council, the Wellcome Trust. In our country, it's the National Institutes of Health and places like the National Science Foundation and the Howard Hughes Medical Institute. That's the place, those are the sources of funding for doing research primarily at academic research institutions, which is where the seminal ideas, the key technologies and key ideas that then later find their applications in practical uses have come from. I don't want to leave the impression that it's a simple process or that it's what we used to think of as a pipeline model where you pump money in at the top to do science and out comes a magical set of applications at the bottom. All you have to do is pump in money at the top and the stuff will happen at the bottom. We know it's not that simple anymore, but there is some truth to the fact that governments all over the world have accepted responsibility for supporting basic research in large part because it doesn't work to leave it to the private sector to do that work because there is no incentive to do that otherwise. So governments all over the world have reached more or less the same conclusion, and that's actually a new policy formulation that only began to take root in World War II in a big scale. This is one of the guys who was most responsible for doing it. This is Vannevar Bush, who was the US head of the war effort in the United States, the Office of Science, Scientific Research and Development. And this is a picture, this is actually not a picture of the ENIAC, this is a picture of a computer that he designed. This is a guy who did design computers before he became President Roosevelt's chief science advisor. But the most famous things that he did are not what he did as an administrator, although those may be the most important things that he did. He, for example, is the person who advocated to President Roosevelt that the Manhattan Project should go forward before it was called the Manhattan Project. He was the key spigot that had to be turned on for the money to flow into that project in the early 1940s. But he's actually known for two products. One is most computer scientists know him for writing this article that he called As We May Think, which many credit with being the first idea of hypertext, where rather than having a trail of linear things, like if you think of a Yahoo menu or a hierarchical menu, this is how you find things. He said, no, the way that the human mind really thinks and the way we should think about designing computers is to be able to find things by figuring out how you got from point A to point B through connections, through nodes, through networks. And his article, As We May Think, was actually his version of a machine that would allow you to do this. Now, for him, the storage medium was actually going to be microfiches. You'd push a button on your desk and it would find the right microfiche. That's because the most high-density way of storing information was photographic film with grains in those days. Now, of course, we would know, no, it's much more efficient to put that in digital bits inside of a digital memory. But that technology didn't even exist in his day. And the other really important thing that he formulated was science policy. As President Roosevelt saw the end of the war coming up, he asked his most trusted science advisor to tell him, what should we do after the war ends? And Vannevar Bush wrote this classic, relatively short document that's called Science: The Endless Frontier, which was a partial template for science policy after the war. The part of it that pervaded thinking after the war was that scientists should be left to their own, that scientists were an autonomous political group that should be trusted to spend money efficiently and pick which projects are most technologically promising. So he argued for scientific autonomy, and he argued for keeping scientists in their academic homes, rather than what had been the habit up until World War II, which was every time we had a war, you would hire a bunch of scientists into government, and they would do all their work as government employees. And that's actually what changed in World War II. After the war, people returned to their universities, and the government continued to fund them. And his argument in Science: The Endless Frontier was basically that is a responsibility of government. So these days, actually, Vannevar Bush, he was what these days would be pretty close to a Tea Partier. He's a pretty conservative Republican guy who hated paying his taxes. He got into a big spat with the Washington, D.C. government when they actually had the temerity to expect him to pay taxes for the place that he was living, because he had a house up in New Hampshire where you live free or die. And you're not supposed to have to pay taxes if you live in New Hampshire. So he actually, he was a very conservative guy who was advising one of our most liberal presidents in American history, Roosevelt. Roosevelt really trusted this guy's advice, and unfortunately, Roosevelt died before he finished his report. The report came out in July, about a month before the bombs were dropped on Hiroshima and Nagasaki. So there is no mention of the single most important technological project during World War II in his report. In fact, the first mention of a technology is penicillin, which up until the Manhattan Project, he considered the signal achievement of the war effort on the technological frontier. So Vannevar Bush comes up with this template of government funds science, and it produces really, really useful things, and industry is also involved. But there's a segmentation, there's a differentiation of functions. My argument here is that, in fact, that worked out pretty well to explain a lot of what happened in these two miraculous technologies, computing technology and genomics. And they've worked their way through a really intricate system that involves government funding, academic research institutions, and industrial firms that do all sorts of different things in really tight coordination, but without much of an overall design. It's kind of a chaotic system. It's an intricate mutualism that none of us fully understands. I'm not going to go into the details, although this is what I love to teach. I'm not going to bore you all with what I love to teach. I want to move on, because I do want to get into the genomics. But this is my last slide about, that refers back to computing, and it's my transition to genomics. Here are two figures that you guys may or may not know about. The guy on the left is J.C.R. Licklider, who is probably the single most important figure in the history of modern computing, at least in my book. He was a psychologist by training, worked at MIT, worked at IBM, worked at MIT, worked at I mean, he shuttled back and forth. He had a vision in the early 1960s that computers were not going to be calculating machines. They were actually going to be extensions of the human mind that would do the things that humans aren't as good at doing. And if you could get a machine to do things like count things, keep track of things, organize things, you would be able to do things with your mind in symbiosis with a machine. And he called it the man-computer symbiosis in a classic article that was published in 1966, but actually he was working on that template for years before that article came out. He became in charge, he was put in charge of a part of the US government that became the fount of almost all of the computing technologies that I've been talking about. What we think about as modern computing, memory, software, networking, all came out of the Information Processing Techniques Office of the Defense Advanced Research Projects Agency, which is part of our military, that was created actually in the Sputnik scare in the United States in 1958. It was created in 1957 as a space agency to adjust for the failure of the United States to get into space first. It was a space agency for exactly 1 year until NASA was created. And then it turned to developing new technologies for the military use, but to do that in a way that wasn't part of the Army and wasn't part of the Navy, because President Eisenhower had concluded that the reason that we didn't get into space quickly was because the Army and the Navy were spending so much time and energy fighting each other that they were forgetting what their overall mission was, which was to get technologies to work. And so he said, I want somebody who answers directly to me to start creating technologies, and then they can farm them out to the military. And that was the origin of the Defense Advanced Research Project Agency. This is the guy who was put in charge of the computer part of that, and that is now a legend in the field of computing technology. I think there's a loose analogy to another guy who's going to now take over the story. This is Fred Sanger, who is still alive, lives outside of Cambridge, where he tends his garden. He got 2 Nobel Prizes. He got his first Nobel Prize for understanding the sequence of insulin, and his second Nobel Prize for inventing one of the technologies for sequencing DNA. He's a really, really humble Quaker guy who is revered at the University of Cambridge as the spiritual father of a way of doing science that is open and focused on structural understanding to explain biological function. And he is one of the pioneers of DNA sequencing. So here are the key technologies that gave rise to what became known as the Human Genome Project. I already talked about the fact that we were developing ways of analyzing DNA faster, better, cheaper. I talked about recombinant DNA and DNA sequencing, but I want to point out that, in fact, none of this would have mattered if we don't throw in this fourth technology of computing. And the development of the Human Genome Project was just as dependent on the development of the World Wide Web and the ability to share information across computing systems very fast as high-energy physics was. This was the scientific shift in gears from one experiment at a time of low-density information to very high-density, high-throughput biology that is driven by data more than it is hypothesis. Now, the old way of doing biology hasn't gone away, and it's still really important. But what has been layered on top of it is a very data-intensive, high-throughput, high-tech, whiz-bang way of doing biology that starts with masses of information. And the place where that's most apparent is DNA sequencing. So here I'm shifting gears to what we now think of as modern genomics. And here is the driver of what's going on in that world. This is a curve that's showing how fast DNA sequencing is evolving as a technology. This is a logarithmic curve. And this slow-seeming graph along here is very much the trajectory that the engineer from Intel, Gordon Moore, described as Moore's Law. That's actually an astonishingly fast pace of technological evolution. Moore's Law was basically, it's stated in different ways in different contexts, but basically Gordon Moore is an engineer, so he does curves. And he observed that basically you can cram the same number of transistors into a smaller space. About every 18 months, you could get twice as many transistors in the same amount of space, which means for the same cost, you can produce twice as much computing power. And that put computing on a trajectory to double its power as a kind of a loose proxy for power every 18 months. And he basically said, you know, this has been going on for over a decade, and I don't see why it should stop. So this is a Moore's Law of doubling in technological power every 18 months. And that's the curve that we were following during the period of the Human Genome Project here, which started right around here, 1985, until 2003, 2004, which is when the reference sequence was published for the human genome. What's interesting is there's an elbow here, and suddenly we're in a hypermoore's curve. And for about 5 years, we had an increase of a full log per year in the speed of DNA sequencing production, which means 10 times as much stuff generated, data being generated each year for a 5-year period. Now that's huge. That's truly huge. And none of us in this room have probably fully felt that yet. The topic of my lecture really is that I think we're going to start feeling it pretty soon. And here's one measure of that. If you think about it, the Human Genome Project was thought to be nearly impossible. That's why it was controversial when the idea was first hatched in the 1980s. One reason was Wally Gilbert, one of the other co-inventors of a sequencing method, stood up in 1986 at Cold Spring Harbor and scribbled on the blackboard, there are 3 times 10 to the 9th base pairs in the human genome. Let's say it's going to cost us $1 a base pair to generate that sequence. Well, that means if we want to do something called the Human Genome Project, it'll cost roughly $3 billion. The scientist sitting in the room thought he was completely insane. Why would you sequence the human genome? Why would you spend $3 billion to create a bunch of A's, G's, T's, and C's, and get them in the right order? Crazy idea. And one of the reasons that it seemed crazy was because it was so damned expensive. Well, and it turned out that that wasn't too far off within one significant figure. That's probably about what it cost to produce the first reference genome. When it was produced in the year 2000, it was refined until 2003. By the time we move to the more modern era, it's now, I'll show you a slide in a minute, but basically jumping ahead, it's now, you can buy your genome for $8,000 from a commercial service. You get your doctor to send some DNA for you to either the company Illumina or Complete Genomics. There are a bunch of companies that are doing this for roughly the same cost of $5,000 to $10,000. But back here, I'll go back here. The point of this slide is also that we haven't finished this revolution. Oxford Nanopore just last month announced that they're going to have a machine they expect by the end of next year that will be able to do basically a genome, and there are 2 or 3 companies are talking about being able to generate a genome's worth of sequence information in less than an hour, 15 minutes to an hour. This is an astonishing pace of technological innovation, and it seems like it actually might happen. Let's say they're off by a year or 2. It's still astonishing to think that it might actually happen. What that means is that we are going to find that DNA sequencing is no longer, it already has stopped in the biological sciences. It stopped being the limiting thing for an awful lot of biological studies. It used to be the most expensive thing that actually limited what you could do if you wanted to study a biological question. It's now disappearing into the mist, and the things that are looming as much more difficult to do are actually interpreting the data and making sense of it and following people over time. And that's where the science is going to go. It's going to go into computation and following people's lives over time and collecting data in a way that allows us to make meaning out of them. It won't be focused on the sequencing technology anymore after the next year or so. So here's another little feature of that kind of a technological innovation. And I'm going to show you a whole bunch of figures about what that means when you're generating data at a certain rate in our space of genomics. This is how much it would cost to generate, what you would do once you had $1 million worth of genomic information. So this is, the constant here is spend $1 million generating sequence data and then put it into a computer. And then what happens to it? So in 1990, if you did that, it cost almost nothing. It cost $10 to store it, and it took 1 second to transmit it. What that meant is if you were running a sequencing machine in your lab, what you would do is you'd hit a button. This is actually what we did. You'd hit a button, and the data would go off to GenBank. And it would go straight into the computer, and it would become part of the database. It only took a second to do that, so it was pretty easy to do that. Well, now, it costs $200,000 to store $1 million worth of data. So the cost of storage is now getting kind of comparable to how much it costs you to generate the data. And it takes several hours to move it. In fact, you'll see in a slide that I show you, a lot of times you don't even move the data through wires anymore. You actually hand people a hard disk because it's easier to pay for an airplane ticket and take it someplace than it is to pump it through the rather narrow channels we have for transmitting information. And the projection is that it's going to cost several $1 million to store the data you can generate with $1 million worth of sequencing reagents by the end of this decade. And we aren't going to be transmitting it. That's simple. The data become immobile. And here's another way of displaying the curves that are beginning to cross. And what it means is we're going to have to be doing things in a different way, and we simply aren't going to be thinking about biology as creating information at one place, moving it to another place where it's going to be analyzed, because that's not going to be possible anymore. The reason being we're generating so much data. And now I'm going to shift gears. I'm painting a picture of lots and lots and lots of data. And now I'm going to shift gears about, now what does this mean in our own lives? Well, here's one aspect of kind of interesting. This is not something that any of us who were talking about the Human Genome Project in the mid-1980s even thought about, frankly. Turns out that genomics got pretty hot in the 1990s as something that you could actually go to Wall Street and get money for. And you have a picture. This is a guy named Bill Hazeltine who became the head of this company called Human Genome Sciences. And he's ringing the bell at the NASDAQ. That's why you see the NASDAQ in the background. Hot new company, makes its initial public offering. He got to ring the bell at NASDAQ to open the market that day. This became one of the companies, and in the bottom left corner here is Randy Scott. These are two companies that were formed in the early 1990s, 1992, 1993 period. Their business model was to do something really simple. Create DNA sequencing from the juicy bits of the human genome. So what do I mean by that? Their idea was, hey, it's becoming possible to identify genes within the human genome. What do? You take the parts of DNA that are transcribed to RNA, edited down to messenger RNA, turn that back into DNA. That's called complementary DNA. It's a piece of DNA that basically corresponds to the coding sequences that make up a protein. And it turned out, these guys said, hey, you know what? What we're going to do with our companies is we're going to identify all the bits of DNA that correspond, that code for proteins. We're going to sequence them in our labs, and then we're going to send those files off to the patent office, and we're going to get patents on those genes. And then anybody who finds something useful to do with one of those genes is going to have to come to us. They're either going to have to license it, or we may find something really valuable to do with one of those genes, and we will have the intellectual property that allows us to do that. That was their business model. And the point of saying this is these are the first two companies that I'm aware of that actually thought about DNA sequencing as the core of their business model. But this wasn't sequencing in the sense of making instruments. This was sequencing in the sense of creating information about individual genes, protein coding sequences of genes, and turning that into money. So that was kind of surprising in the 1980s, and it turned out that commercial genomics became a pretty big thing. I'm not showing you any of the slides, but basically, at one time, one of the startup companies was valued at the same level, Celera Genomics, the company that competed with the Human Genome Project that no longer exists. It had a very sharp trajectory. At one point in its trajectory, it was valued higher than General Motors before it crashed and burned. And so it became really big business, but it also had a bubble and it disappeared. And I won't go into all the history of that, except I do want to bring it back to what it means for us in our own lives and where the world might be going. This slide shows you the two people whose genomes as individuals were first done. And you'll see that they were done within a few months of each other. And you may even recognize the names. This was the era of celebrity genomics. You have Jim Watson in his dapper white suit and blue tie. Gorgeous, gorgeous outfit that day. And this is the team of folks who gave him his genome on May 31st of 2007 in Houston. And the guy on the right with the blue tie with no suit on is Jonathan Rothberg. He is the inventor of two of the technologies for DNA sequencing, ion torrent and 454 sequencing. And Richard Gibbs is the guy standing right next to Jim. He's the guy who ran the Genome Center at Baylor. The only woman in the picture is Amy McGuire. She had the unenviable job of trying to talk to Jim Watson about the ethical implications of sharing his genome on the internet. So she's the one who got to ask him what it meant for his family and the other people in his family if he shared his genome. This, of course, is Craig Venter in his nautical splendor. wearing his suit. He's a big sailor. This is from the cover of his book called A Life Decoded. His genome was published and then put on the internet in an open source journal in October of 2007. So notice, we have a reference genome on the human that has been announced by a bunch of politicians in June of 2000, but not really complete. It's complete by 2003. And then 4 years later, we have two individual people who have had their entire genome sequenced. At more or less that same time, we have the beginning of a consumer movement that is beginning to pay attention to what's going on with these technologies. And you have the origin of these two companies within a month of one another. They're both based in Silicon Valley. They're both funded by capital sources that you would know the names of. In the case of Navigenics, this is the same group of investors that gave rise to companies like Genentech. It's Kleiner Perkins and Byers and Caulfield. It's probably the single most prestigious venture capital firm on Sand Hill Road outside of Stanford's campus. And down here at the bottom, you have the origin of the company 23andMe. This is Anne Wojcicki. Anne's older sister, Susan, is the person from whom Sergey Brin and Page rented the garage, quite literally, to start up Google. And Sergey married Anne, and she got really interested in an idea that this woman, Linda Avey, had about applying the newly ripening technologies of genomics and selling information to people directly to get information about their own genomes. So that is their business model. Both of these companies still exist. They're changing their business models in real time, but the fundamental idea is don't get between me and my genome. Now, these companies are somewhat different. Navigenics is a little bit more of a medical model, and they always have had genetic counselors and access to physicians. 23andMe is unabashedly and unashamedly direct-to-consumer, don't get between me and my genome. Very strong kind of web-based ideology. Not to be left out, Kari Stephenson, who's not too distant a neighbor. His company Decode was based in Iceland and was originated in order to take advantage of the information resources that were made available through a long debate in the Icelandic political system. Rather controversial. It's a company that's been wildly successful at identifying genes that are associated with diseases of various kinds. It's been far less successful in finding a business model that will make money for its investors. So it's had kind of a shaky business history, but it's a really, really good scientific company. And they were using more or less the same technologies as these other two companies, and they jumped into the fray of direct-to-consumer genomics. It's not clear they're going to stay in that game. And then at the bottom of this slide, we have two other things that are becoming a new aspect of this social movement. You have George Church, who's the guru of the Personal Genome Project. He's a guy who worked in Wally Gilbert's lab. He actually flunked out of graduate school at my university, Duke, and he shows this I flunked out of Duke slide in his presentations quite proudly. He ended up going and working for a Nobel laureate and working on sequencing technology, and he was one of the co-founders of one of the companies that went the next step and said, you know what, we will sequence your genome. And notice the price. In November of 2007, they were saying it's a bargain at $350,000 to get your genome. And I earlier told you you can now buy it from Illumina or Complete Genomics for $8,000. So that's a difference between 2007 and 2012. Over here on the right are 8 people who are participating in a social experiment of releasing their genomic information into the public. These are folks who went in, signed a very elaborate, informed consent that basically said, we are going to be information altruists. We're going to share all this information about our genomes because we want to see what happens when you do that. And one of them in the green shirt here is my colleague, Misha Angrist, who works right down the hallway from me and has written a book about it called Here is a Human Being. So just to mention a few things about what's going on in our science. I mentioned that it's a lot of data. We're just creating reams and reams of data. And I showed you the first 2 people who had their individual genomes sequenced. Now, how many people have had their genome sequenced in the interim? That was 2007, so that was less than 5 years ago. Actually, I don't think anybody knows how many people have had their genome sequenced, but it's almost certainly more than 10,000 and less than 100,000. So we're somewhere between 10 to the 4th and 10 to the 5th people who have had their genome sequenced. But if you think about it, what's going to happen over the next few years? What I tell my freshman students is they're almost certain by the time they graduate from Duke in 4 years, they're almost certain to have a choice in front of them that they will be able to get their genome sequenced for the cost of their bike. Now, they may not choose to do that, but it's going to be a decision that they make that is in that range of economics. And that rather profoundly changes the likelihood that that's actually going to happen in their lifetime, because what it means is they only need to have a use for sequencing their genome once in their lifetime in order to invest in creating that information the first time. And then, once they've created that information for the rest of their lives, they'll be reinterpreting it. That's a very, very, very different dynamic from thinking about a genetic test that you go to only if you're trying to figure out whether you've inherited the gene that's associated with high risk of breast cancer, or Alzheimer's disease, or Huntington's disease. This is a storehouse of information about a human life that doesn't tell you what's going to happen in your life, but it's going to tell you some things that matter to you in your life that you are likely to want to access over time. And the game is not going to be generating the information in the first place anymore. It's going to be, how do we interpret it? And you're going to have to be following science over time. That's a change in how we think about genetic information. That's one change. The other thing to notice is that information, if I have my genome information on a disk, what can I do with it? Well, what I can do with it right now, for the most part, is I can float the disk and we can play Frisbee with it. That's about it. It gives me no information that I care about in my life, unless I happen to have a mutation that's associated with a disease or something else, or if I want to know whether I'm a Pict or a Viking, I may be able to send it off to a service and figure out who my lineage is related to and all that. But even that is a little bit shaky right now. It's going to take 10 or 15 years before we have really good data sources. But think about it. This is kind of equivalent to the same situation that I faced when I was acting as a secretary in 1979, and I would take an 8-inch floppy disk and put it into something that was called a newfangled thing. It was called a word processor, that allow you to use a computer to do what we used to do by typing things by hand. So you didn't have to type your paper de novo if you made a mistake. You could actually just change a digital bit and it would print out the right way. Profoundly important technology that was really good in allowing me to not have to retype my papers. But the problem was I really couldn't share it with anybody. I couldn't share that information. I couldn't share that file. But guess what happened over time? Suddenly, once there was standardization and sharing of information and being able to transmit that information from one system to another in a network, well, suddenly, I can transmit my paper now, and I've done this several times already today. I send the digital bits someplace, it comes back to me, I do something with it in my computer, the other person can use it and I can use it, and it's interoperable. This is an incredibly powerful change in how you handle information. And what it means when you're thinking about the questions that we want to raise about the human genome, I've talked about medical illnesses. What I want to know is, what's my risk of getting this disease or that disease? How are we going to find that out? What we're going to have to do is we're going to have to study thousands, hundreds of thousands, millions of people over time, because the likelihood that any given person is going to develop a disease tomorrow is quite low. But if you do that for millions of people over time, you're going to have a way of finding out who develops which diseases over time. And if you can track that information over time and pool the information among millions of individuals, suddenly you have a powerful way for figuring out what's getting broken in a human body if you're keeping track of a lot of information. But you need to be able to keep track of everybody's information over time. You need a network technology that allows you to do that sort of thing. That's what we actually don't have the infrastructure for doing right now. That's why I showed the slide of Steve Jobs and Bill Gates. Because what they did is they developed companies that took ideas about how to use technology, how to develop technology, and turn it into something that people really wanted to use in their daily lives. My hypothesis is that we're probably going to figure out all sorts of uses of genetic information that matter in our lives. And we will have to come up with the structure that allows us to share information among ourselves in ways that feel safe to ourselves. Now, here's another place where genomics is going to be different from computers. Genomics is going to be information about you, and you may not want to share all that information publicly. I have not had my genome sequenced, and if I had, I wouldn't be sharing it on the internet. The reason is because I don't trust the world enough to share all of my genetic information, because I think somebody might misuse it someday. I think, you know, suppose I'm at high risk of Alzheimer's disease. I don't know that right now, but I might have the ApoE4 allele. And once I have that, it could be that somebody's going to say, no, Bob, you can't buy long-term care insurance anymore. Or if you buy it, you have to pay a higher rate. I don't want them to know that. I certainly don't want them to know that before I do. So I don't want this to be shared until I've got some control of that information. So the information about our genomes is not going to be as easily transmissible as a word processing file. We're going to have to come up with a safe way to share lots of information. So I talked about the medical stuff. We're going to have to follow lots of people over time. I'll finish with just talking about, let's talk about this other thing that might be something that we actually are really, really curious about. And that's the study of ancestry. The book that I showed the picture of it actually seems pretty likely that we're going to be able to follow lots of human lineages at a much, much higher level of resolution than we've ever been able to do before because of genomic technologies. And I'm going to get at that by showing you this incredibly, incredibly interesting slide. This is a slide from a paper that was published in Nature in 2008 by John November from UCLA and Carlos Bustamante, who at that time was at Cornell. He's now at Stanford. This is an incredibly simple thing that they've done here. What you see is a picture of Europe. You probably recognize it there in the background. What they did was they projected that map of Europe and did an incredibly simple-minded thing. They took differences that you can detect using off-the-shelf genomic technologies. You look for differences that are quite common among human beings, and you select out the ones that are most useful for figuring out ancestry-informative markers. You look for variations that tend to be different between different populations who live in different places. You don't need to know what those things mean. You just notice that people in one place have a higher frequency of having this spelling of this piece of DNA than the people who live over here. You don't know what it means. You just collect it. You throw it into a computer. And you say, computer, tell me, on this axis, separate all the markers that we found most informative on this principal component. And in this direction, calculate it on these other sets of markers. And you throw it into the computer and say, computer, figure it out. And this is what the computer did. So what you've got is a set of markers on two axes, two principal components. And all they did is throw it in the computer and say, map it in. Look what happens. You probably can't see it very well, but you'll see the blotches. What they also did is they took samples from people, and they said, where are you from? Where are your parents from? Where are your grandparents from? And what they discovered is if that's all they did, they could predict within 700 kilometers where 90% of the people that they sampled who could answer that question. My parents, my grandparents came from Italy. My parents, my grandparents came from Germany. And that's what they've done. They've taken national origin data and thrown it onto a map. And all they did was tell the computer to organize it according to these two origins. It's an absolutely astonishing indication of the kind of information that is stored in our genomes that we don't think of as being stored in our genomes. This doesn't tell us anything about genetic determinism or who's smarter or dumber or anything like that. It doesn't tell us any of that stuff. What it does tell us is that when people have lived in the same geography over a period of time and you look at markers that tend to be geographically dispersed, then you can actually start creating maps like this. And we have a technology, therefore, for kind of figuring out human lineages that is quite powerful and quite granular. So there are another example of this same kind of science that's going on is there's a tribe in Puerto Rico that no longer exists. There's nobody from that tribe that exists on the planet anymore. But a bunch of scientists were invited to Puerto Rico and asked, gosh, could you kind of figure out what the genome of this lost tribe looked like by taking genomes of people who live in Puerto Rico now and identify the bits of their genomes that they've inherited from people who used to live in Puerto Rico and no longer, their tribe no longer exists? Well, yes, it turns out, yeah. You can reassemble the genome of a lost population on average by taking the DNA from people who are still living in that geographical area. Now, there are lots and lots of complexities. There's going to be a lot of social mischief that's associated with interpreting data like this, because it's going to be really easy to overinterpret the fact that you can make geographic associations and not fully understand the science. But the point is, we're going to have to get used to it, because the technologies are there. Genomic information is going to contain information about who's related to whom over time. And to the degree that that is related to where people live and how people live, we're going to be able to begin to figure it out. And we're going to be able to make correlations by using computational methods applied to highly granular genomic information. So, a few things to notice about what that's going to mean for us that we're going to have to contend with, and I'll finish on this slide. One is genomic information. There is this long tradition about what's special about genomics, what's special about genetics. There's the idea of genetics exceptionalism. All of us in the world of bioethics have this tendency to say, gosh, there's something special about the human genome, and it's not just because we get paid to think about it, we actually think there's something special about it. Well, turns out, most of the time, if you really think about it, there's nothing really so special about DNA or genetics. We've confronted problems, social problems in one context or another, and we can usually reason our way to a policy solution by analogy to things that we've known from the past. But there are a few things that are distinctive that we do need to be paying attention to them. One is the fact that, as I alluded to when I showed the slide of Jim Watson, when Jim Watson decided to share his genome, he wasn't just sharing his genome, he was also sharing information that people around the world, by looking on the internet, can infer information about his son, both of his sons, Rufus and Duncan. So he was making a decision not just about himself. He was making a decision that also influences other people in his family. And that's going to be true. When you're making a decision about sharing genomic information, you're making a decision that also affects other people in your family in a much more precise way than some of those family members may like. So that introduces a true complexity. It's not completely unique to genetics, but it's unusual. It is also the case, I've already alluded to this, that genomic information is going to feel more private. I've already said I don't want to share my information with the world until I feel like it's safe to do so. And I don't know when I'm going to know. Maybe never. That may never be the case. We don't know yet what the harms are going to be. And the only way we're going to find them out, probably, is to discover them by trial and error. That's how policy usually works. We react to catastrophe. And that may be, unfortunately, how we figure out what the problems are going to be arising out of genomic information. And the last 2 points are to point out that, in fact, we now have the technologies for generating lots of information, but we don't have the infrastructure or the technologies for figuring out what it means. and we don't have the social networking capabilities that are going to allow us to do that efficiently yet. So that's the hard work. That's going to be a whole series of questions about how we share information safely among ourselves. And at the very least, if you even take it in these two very simple-minded contexts that I framed, medical uses of information and ancestry information, we actually don't have very reliable interpretive services even now, despite this 5 decades of spending tons of money on molecular and cellular biology. What are we missing? We're missing all the data about all the people over time. And that hasn't gone away. That's going to be the problem that needs to be solved if we're really going to make sense of it. And before you can make your choice here between whether you're a Pict or a Viking. So, thank you.
This production is brought to you by the University of Edinburgh.